All right, welcome. What we're going to do here is build a solid, clinically focused framework for classifying the non-Hodgkin lymphomas. And we're going to concentrate on the B-cell malignancies because, well, they make up the vast majority of these diseases.
So let's jump right in. The term non-Hodgkin lymphoma, or NHL, it's not one single disease, not even close. It's actually this huge umbrella term for over 60 different types of cancers of the lymphatic system. So the central problem for doctors and pathologists is, how do you bring order to that chaos? How do you classify them in a way that actually tells you something useful about how they'll behave and how to treat them? The answer isn't just memorizing a long list. It's about understanding a really fundamental biological journey. And here it is. The real key, the organizing principle for almost all B-cell lymphomas, is the exact stage of B-cell development where things go wrong, where that cancerous transformation happens. If you can map a lymphoma back to its original cell type along this developmental path, you can start to predict its genetics, its characteristics, and how it's likely to play out for the patient. And the main stage for all this action is the lymph node's germinal center. So this is our roadmap. We can basically break down the B cell's life into three big phases. First, you've got the pre-germinal center stage. These are the naive B cells, just waiting for instructions. Second, the germinal center itself. This is a super dynamic place where B cells go through massive changes, multiplying as centroblasts and then maturing into centrocytes. And third, the post-germinal center stage. Here, the cell leaves as either a memory B cell that hangs out in the marginal zone, or it becomes a plasmablast on its way to making antibodies. Every single one of these stages is a point where a malignancy can arise. Okay, let's put this framework into action, starting right at the beginning of the path. The first major lymphoma we run into comes from that pre-germinal center stage, specifically from those naive B cells that live in the mantle zone, right around the germinal center. And this is the origin of mantle cell lymphoma, or MCL.
Let's look at the molecular fingerprint of MCL. You'll see it expresses CD5 and cyclin D1. Now that cyclin D1 is the real problem. It's a key that controls the cell cycle, and it's massively overexpressed. Because of MCL's signature genetic mistake, the T1114 translocation. What happens is the cyclin D1 gene on chromosome 11 gets moved right next to the super powerful immunoglobulin heavy chain promoter on chromosome 14. B cells are designed to crank out immunoglobulins, so that promoter is always on full blast. Now it's mistakenly driving cyclin D1, leading to runaway cell division. Interestingly, a key prognostic factor is a protein called SOX11. You'd think it would be bad, but in the typical nodal form of MCL, having SOX11 is actually linked to a better response to treatment. The less common leukemic version of MCL, on the other hand, is usually SOX11 negative.
Now we enter the germinal center, and this is where a really critical split happens. You've got two cell types here. The more mature, quieter cell, the centrocyte, is what gives rise to a typically low-grade, slow-growing cancer, follicular lymphoma. But the other cell, the highly proliferative, less mature centroblast, well, that's the cell of origin for two of the most aggressive high-grade lymphomas we know, diffuse large B-cell lymphoma and Burkitt's lymphoma.
So here's the profile of that classic low-grade lymphoma. Follicular lymphoma comes from centrocytes. Its markers, CD10 and BCL6, basically scream, I'm a germinal center B cell. The defining genetic event is the T1418 translocation. This moves the BCL2 gene from chromosome 18 over to that powerful immunoglobulin locus on chromosome 14. Now, BCL2's job is to prevent cell death. It's an anti-apoptosis protein. When it's overexpressed, it basically makes the cancer cells immortal. They just refuse to die. The biggest worry with this otherwise slow-growing disease is the risk that it can suddenly transform, what we call a Richter transformation, into a much more aggressive high-grade lymphoma, usually DLBCL.
Okay, as we exit the germinal center, we find two more points of origin for cancer. B cells that mature into memory cells and settle in the marginal zones can turn into marginal zone lymphomas, like maltoma. And then, those cells that go down the plasma cell path, the plasmablasts, they're the source of a very specific and much tougher subtype of DLBCL, called the activated B cell or ABC type.
Maltoma, which stands for mucosa-associated lymphoid tissue lymphoma, is the classic example of a marginal zone lymphoma that happens outside the lymph nodes. It's a low-grade cancer that's really driven by long-term chronic inflammation. The textbook case is gastric maltoma, which is directly caused by a chronic Helicobacter pylori infection. In fact, if you get rid of the bacteria, the lymphoma can actually go away on its own. You see the same thing in Sjogren's syndrome, where chronic autoimmune inflammation in the salivary glands sparks the maltoma.
Some of these cases have a T1118 translocation, which creates a fusion protein that keeps a key inflammatory pathway, NF-kappa B, switched on. Okay, so we've established our biological roadmap. Now we can overlay a second, equally important classification system. This one is based entirely on clinical behavior, basically how aggressive the cancer is in a patient. This grading is absolutely critical for figuring out prognosis and deciding how urgently and intensely we need to treat it. And here's how they sort out. On the low grade or indolent side, we have things like small lymphocytic lymphoma, which is essentially the solid tumor version of chronic lymphocytic leukemia, or CLL, plus follicular lymphoma and marginal zone lymphoma. On the aggressive, intermediate to high-grade side, we find diffuse large B-cell lymphoma and Burkitt's. Now, look where mantle cell lymphoma is. Even though it comes from a pre-germinal center B-cell, clinically it acts aggressively and it's tough to treat, which is why it belongs over here with the high-grade diseases. So let's really zoom in on those two major high-grade B-cell lymphomas. Getting a handle on the unique features of DLBC and Burkitt's lymphoma is essential because they represent the most common and the most aggressive phases of NHL. DLBCL is by far the most common type of non-Hodgkin lymphoma. But as we've seen, it's not one single thing. We've identified two main subtypes based on their cell of origin: the germinal center B-cell type, or GCB, and the activated B-cell type, or ABC. That ABC type comes from a post-germinal center plasmablast, and unfortunately, it carries a much worse prognosis. In the lab, pathologists use something called the Hans algorithm. It's a panel of stains to tell them apart. They use markers like CD10 and BCL6 for germinal center origin, and MUM1 to point to a post-germinal center origin. Clinically, it's also really important to note that DLBCL is often an aids to finding illness.
Now, within this group of aggressive lymphomas, there's a genetic subtype that is particularly feared. We call it a double-hit lymphoma. This is where the cancer has a genetic rearrangement of the super powerful mice oncogene plus a rearrangement of either BCL2 or BCL6. You're getting hit with two major cancer-driving events. And if all three, mice, BCL2, and BCL6 are rearranged, that's a triple-hit lymphoma. As you can imagine, both of these have a very poor prognosis and are incredibly difficult to treat. So if DLBCL is the most common aggressive lymphoma, Burkitt's is the poster child for aggression. It's recognized as the fastest growing human tumor, period. And this incredible growth rate isn't just random. It's the direct result of one single defining genetic problem: the complete and total dysregulation of the c-MYC oncogene. And this number right here says it all. K67 is a marker for cell proliferation. In most cancers, you might see a K67 index of, say, 20 or 40%. In Burkitt's lymphoma, it's approaching 100%. What that means is that at any given moment, basically every single cancer cell in the body is actively dividing. That's why it presents so explosively.
So let's quickly recap the key features of Burkitt's. Their genetic hallmark is that CMYC translocation, usually T814, which puts MyC under the control of that powerful immunoglobulin gene. Under the microscope, it has this classic starry sky look. The sky is a dark blue sheet of lymphoma cells, and the stars are pale macrophages that are literally eating up dead tumor cells, which tells you just how incredibly fast the tumor is turning over. Clinically, you always have to worry about it spreading to the central nervous system. And finally, there are two distinct types: the endemic form, seen in Africa, which often involves the jaw and is almost always linked to the Epstein-Barr virus, and the sporadic form we see elsewhere, which usually starts in the abdomen. Let's pull the lens back one more time now and think about some of the external factors that can cause these lymphomas, and also talk about the universal language we use to describe how far the disease has spread. This table really drives home the powerful link between infections and lymphoma. The Epstein-Barr virus, EBV, is a huge player. It's tied to endemic Burkitt's, some types of Hodgkin's, and lymphomas that happen after organ transplants. The severe immune deficiency from HIV basically takes the brakes off the immune system, allowing aggressive lymphomas like DLBC and Burkitt's to emerge. We've already covered H. pylori and Multoma. Other key links you need to know are human herpes virus 8, or HHV8, with primary effusion lymphoma, and hepatitis C with certain marginal zone lymphomas. Okay, so once you have a diagnosis, the next critical step is staging, finding out where the cancer is. And the universal standard for this is the Ann Arbor staging system. It's pretty logical. Stage 1 is just a single lymph node region. Stage 2 is 2 or more regions, but all on the same side of the diaphragm. Stage 3 means there are involved nodes on both sides of the diaphragm.
And stage 4 means the cancer has spread widely into organs outside the lymph system, like the liver or bone marrow. Beyond the specific type and stage, doctors look at a few other key factors to get a better sense of the prognosis. These are the big unfavorable ones: being older than 60, having a high level of serum lactate dehydrogenase, or LDH, which is a sign of high tumor burden, and having a poor performance status, which is a measure of how well a patient is functioning, are all bad signs. The same goes for having cancer in more than one site outside the lymph nodes, and of course, having an advanced Ann Arbor stage of 3 or 4. These all get plugged into scoring systems to predict outcomes. Now, it's really important to make one final point. We've spent this entire time talking about B-cell lymphomas because they make up about 85% of all NHLs. But you have to remember there's a whole other group of T-cell lymphomas out there. They are a complex group of their own with different classifications and behaviors and include diseases like anaplastic large cell lymphoma and mycosis fungoides, a lymphoma of the skin. We don't have time to dive into them today, but you need to know they exist. So, to wrap it all up, here's the bottom line. This huge, confusing list of non-Hodgkin lymphomas isn't chaotic at all. You can make sense of it. By using these overlapping frameworks, by mapping a cancer to its B-cell origin, layering on its clinical grade, and understanding its specific genetic drivers, you can turn that simple list of diseases into a powerful, logical, and interconnected system that actually helps you take care of patients. And this really leaves us with a fascinating question for the future. For more than 100 years, we've classified lymphomas based on what the cells look like under a microscope. But now, with genetic sequencing becoming routine, we're seeing the complete molecular landscape of these tumors. So the question is, are we on the brink of a massive shift? Are we going to move away from classifying cancers by how they look and towards a new system based purely on a tumor's fundamental genetic signature? It's an exciting time, and honestly, only time will tell.